everyone, our topic is the determination of vitamin C in fruit juice by volumetric analysis. But first, let me introduce myself. My name is Nofazan Nabi Zulkifli.
And my name is Nur Nawara Wadiha binti Abdullah.
My name is Muhammad Haziq bin Razai Azrul Anwar.
And my name is Nuri Marina Akila binti Jasmine.
First of all, vitamin C, also known as ascorbic acid, is a water-soluble vitamin commonly found in fruits and vegetables, especially citrus fruits such as oranges and lemons. It is an important nutrient required by the human body. Vitamin C also functions as an antioxidant that helps protect body cells from damage caused by free radicals. It also supports the immune system, helps in collagen formation, and aids immune healing. The determination of vitamin C in fruit juice is important because different fruit juices contain different amounts of vitamin C. The concentration may vary depending on the type of fruit, processing methods, and storage conditions. Exposure to heat, air and light during storage can reduce the vitamin C content in juice. Therefore, analyzing the amount of vitamin C helps evaluate the nutritional quality of fruit juice products. This analysis is also important in the fruit industry to ensure products' quality and provide accurate nutritional information to consumers. Next is volumetric analysis, which is a quantitative analytical method used to determine the concentration of a substance in the solution through titration. In this experiment, vitamin C in fruit juice reacts with the standard solution until the endpoint is reached, allowing the concentration of vitamin C to be determined.
Hi everyone, so let's move to the titration. In this video, I'll explain how titration is used to determine vitamin C concentration. As we know, titration is a chemical method used to determine the concentration of an unknown solution by solely adding a solution of known concentration until the reaction is complete. In this experiment, vitamin C, also known as ascorbic acid, acts as a reducing agent and reacts with iodine in a redox reaction. This reaction allows us to determine the amount of vitamin C present in a sample. Vitamin C easily loses electrons and is oxidized into dihydroascorbic acid, while iodine gains electrons and is reduced to iodide ions. Starch is used as an indicator because it turns into dark blue-black when excess iodine is present. The reaction ends when all the vitamin C has reacted and excess iodine remains in the solution. For the titration, the solution appears colorless or light yellow. During titration, the ionic color disappears because it reacts with vitamin C. At the endpoint, a permanent blue-black color appears, indicating that all the vitamin C has been oxidized and the titration is complete. In the food industry, vitamin C determination is used to measure the vitamin C content in fruit juice, soft drinks, and fortified beverages. It also ensures accurate nutritional labeling, monitors product quality, and detects vitamin C degradation during storage. Lastly, the applications in pharmaceutical industry. In the pharmaceutical industry, titration is used to determine the vitamin C concentration in tablets, syrup, and supplements. These methods help verify the level amount of vitamin C, ensure product safety and effectiveness, and identifies underdosed or degraded products.
Slide shows the apparatus and chemical used in this experiment. The burette was used to deliver the iodine solution accurately during titration, while the pipette was used to measure the fixed volume of fruit juice sample. The conical flask was used to hold the sample during titration, and the volumetric flask was used for sample preparation and dilution. The chemical used were fruit juice samples, standard iodine solution, starch indicator, distilled water, and standard ascorbic acid solution. Iodine reacts with vitamin C in the fruit juice, while starch indicator helps identify the endpoint by producing a blue-black color. First, the fruit juice sample was prepared and transferred into a conical flask. A measured volume of the sample was taken using a pipette. A few drops of starch indicator were added, and the sample was titrated with iodine solution from the burette. The flask was swirled continuously during the titration. The endpoint was reached when a permanent blue-black color appeared. The volume of iodine used was recorded, and the experiment was repeated for consistent results. For the titration, the fruit juice sample was prepared properly. The sample was filtered if necessary, remove bulk and solid particles. A fixed volume of fruit juice was measured and diluted with distilled water when required. The solution was then mixed thoroughly to ensure a uniform sample before analysis. This slide shows the titration setup. The burette containing iodine solution was clamped vertically on a retort stand. The conical flask containing the fruit juice, sample and starch indicator was placed beneath the burette. Iodine solution was added slowly while continuously swirling the flask. The endpoint was identified by the appearance of permanent blue-black color, indicating that all the vitamin C had reacted.
Good morning everyone. Today I'll be presenting the results, example calculation and the interpretation of result for the determination of vitamin C in fruit juice by volumetric analysis. In this experiment, the analyte was the vitamin C present in the fruit juice, while the titrant was the iodine solution added from the burette. The average volume of iodine used were 12.5 ml for fresh orange juice and 8.2 ml for packaged orange juice and 5.6 ml for apple juice. Titration: iodine reacted with the vitamin C or ascorbic acid in the fruit juice. The endpoint was reached when a permanent blue-black color was appeared after the addition of starch indicator and remained for about 30 seconds. This indicates that all the vitamin C has reacted and excess iodine was present. For the example calculation, the fresh orange juice sample required about 12.5 ml of iodine solution. Using the conversion factor provided, the vitamin C content was calculated by multiplying the volume of iodine used by the conversion factor. This gave a vitamin C content of 12.5 mg for the fresh orange juice sample. The same calculation method was used for the other fruit samples, resulting in 8.2 mg of vitamin C in packet orange juice and 5.6 mg in apple juice. Based on these results, fresh orange juice contained the highest amount of vitamin C or the ascorbic acid, followed by the packet orange juice, and then lastly the apple juice. Since vitamin C reacts directly with iodine, a larger volume of iodine used indicates a higher vitamin C content. When comparing fresh orange juice and packaged orange juice, the fresh orange juice contained higher vitamin C or ascorbic acid content. This is likely because vitamin C can be lost during processing, storage and exposure to heat, light and oxygen. Therefore, fresh orange juice generally retain more vitamin C or the ascorbic acid than packaged orange juice.
In conclusion, volumetric analysis is an effective method for determining the concentration of vitamin C in fruit juice. This experiment shows that different fruit juices contain varying amount of vitamin C with many factors such as storage conditions, processing methods, and fruit type. Overall, this method is useful in evaluating the nutritional quality of these products and nutritional information for consumers.
Thank you for watching. Please like and share.
Have you learned already? Please comment.
Bye.